Okay, in this video I'm just going to go over the entire practice test that I created and hopefully also provide explanation to all of the problems. And I guess it's a little bit of a test for myself to see if I can get 100 on the test that I created. So let's go for it. Okay, most chemical reactions with a delta G less than 0, so delta G is referring to the Gibbs free energy of a chemical reaction. So when the delta G is less than 0, it's an exergonic reaction, meaning it's releasing energy. Delta G that's positive means that it is taking in energy and it's endergonic. So, important thing to realize is that all reactions with a delta G less than 0 are spontaneous and exergonic. So it's not endergonic, and it has nothing to do with activation energy. And we know that delta G less than 0 means spontaneous. Now, these reactions can also take a long time to complete. If you remember in class, the professor was saying that reactions without the assistance of enzymes would still take place, but they would take a very long time to complete. And that actually works out to the benefit of us. We don't want a bunch of reactions going off in a haphazard way inside our body. So the answer is C, take a long time to complete. Which of the following processes occurs when homologous chromosomes cross over in meiosis 1? The answer to this question is segments of non-sister chromatids are exchanged. So you got to remember, when crossing over occurs, it's non-sisters that exchange, not sister chromatids. Where does photosystem 2 get its electrons? Well, it takes in water and it splits apart that water into oxygen and releases it, and takes in the electrons, and the remaining hydrogen goes into the thylakoid space. So it is water. From which component of light-dependent reactions does NADPH form most directly? That would be photosystem 1. It's not photosystem 2, because photosystem 2 is where the water comes in and the electrons enter. So after photosystem 2, those electrons get moved over into photosystem 1, where they are re-energized and used to reduce NAD plus into NADPH. What is the most common enzyme on the planet? It is Rubisco, because all plants have it. Some of these are not even enzymes. Actually, I don't think any of these are enzymes except Rubisco. So, yeah, all plants have Rubisco. It's used in the Calvin cycle. What do photons do in the thylakoids? Well, they hit the chlorophyll and they excite electrons. Which of the following processes do sister chromatids separate from each other? Well, that happens in mitosis, we know that. And we also know that homologous chromosomes separate in meiosis 1, and sister chromatids separate in meiosis 2. So it would be mitosis and meiosis 2. An algae would be considered a, this one's pretty easy, it's just a photoautotroph. gets its energy from the sun. Which of the following would be less effective when the concentration of substrates is extremely high? This one is actually competitive inhibition, because competitive inhibitors bind to the active sites of enzymes and basically stop them from doing what they're supposed to. However, when there's a ton of substrates, then the amount of inhibitors is not as important, because there's so many substrates, and the chances of one of those inhibitors actually being able to attach to an enzyme is extremely low when the concentration of substrates is extremely high, whereas these other forms of inhibition are not affected by the concentration of substrates. Okay, dinitrophenol is an uncoupler that makes the inner mitochondrial membrane leaky to protons. What effect would this have on ATP production? So when the inner mitochondrial membrane is leaky to protons, remember, we're trying to get protons on one side of the membrane, specifically outside the membrane, to create a gradient. And that gradient is what produces ATP. But if those protons are just leaking right back through the membrane, then you're not really producing ATP, are you? So yeah, it would be greatly reduce ATP production. Without enzymes, reactions would slow down. Enzymes don't cause things to release more energy. It doesn't have anything to do with whether reactions are spontaneous or not. And without enzymes, reactions would still occur, but they just take a very, very, very long time. Approximately how many molecules of ATP can one molecule of glucose produce? That's just 32. Takes 32 ATP, or rather glucose can make 32 ATP through the process of cellular respiration. At metaphase 1, homologous chromosomes are connected only at what structures? That would be chiasmata. That's where they are connected. They're not connected by microtubules or kinetochores or I'm not actually really sure what recombination nodules is, but I can tell you that's not the answer. Chiasmata. And remember, this is during meiosis. What is the bond that links the molecule required to jumpstart glycolysis? Remember, glycolysis requires an initial investment of ATP to get it started. And what is the bond in ATP? It's a phosphoanhydride bond. Which of the following serves as an electron carrier in the electron transport chain? None of these are electron carriers except ubiquinone. It serves as like a relay along the electron transport chain. What actually absorbs photons from the sun? It's a tricky question, but it's chlorophyll. Chlorophyll is the actual thing that's absorbing the photons. Yes, photosynthesis takes place in thylakoids and chloroplasts, but chlorophyll is what takes in the photons. What type of life cycle has both a haploid and diploid multicellular stage? That would be alternation of generations, because it'll go back and forth, haploid, diploid, haploid, diploid. What is an oncogene? An oncogene is basically a proto-oncogene that has mutated to become an oncogene. and essentially causes the cell to replicate out of control. So it's a gene that causes a cell to become cancerous. When a organism is in a state of complete equilibrium / entropy, it is most likely dead. Remember, our bodies are in a constant war with equilibrium and entropy, and when that war wins, we're dead. Cancer cells do not abide by, well, I can tell you one thing, everything abides by the laws of thermodynamics. It is cell cycle checkpoints. They don't care about checkpoints. How much ATP and NADH does glycolysis produce in net? Well, remember that glycolysis takes 2 ATP to get started, and we get 2 glyceraldehyde 3-phosphates, which get oxidized to produce 2 NADH from 2 NAD+, and we also get 2 more ATP in step 7 of the cycle. I don't exactly remember the name of the step, it's not important. And we also get another 2 ATP on the final conversion to pyruvate. So 4 ATP minus 2 and 2 NADH. That is 2 NADH plus 2 ATP. Where in eukaryotic cells does the Calvin cycle take place? That would be the chloroplast stroma. Remember, the Calvin cycle does not require light. During which of the following phases of meiosis do centromeres split and sister chromatids migrate to opposite poles of the cell? Well, we know when they split, it's an anaphase, and we know that sister chromatids has to refer to anaphase 2, because if it was homologous chromosomes, we'd be talking about anaphase 1. So we're actually here, the answer is anaphase 2. The universe wants more of what? Entropy. Entropy measures disorder, and the universe always wants more. The Calvin cycle requires how many molecules of CO2 to generate 1 molecule of glucose? That would be 6. Because remember, glucose, the formula for glucose is C6H12O6. C6 means 6 carbons. What is the humpy? If you're in Dr. Clark's class, you know what humpy means. Anyway, what does it mean in a chart of Gibbs free energy versus time? If you're not in Dr. Clark's class, you may not understand this, but it's referring to the big hump on the chart of Gibbs free energy and time. That is the activation energy. It's like a barrier that must be passed for the reaction to actually take place. And enzymes reduce that activation energy. What proteins act at the G1 checkpoint and can halt cell division? They are RB, P53, and P21. This is in the module text. It may not have been covered very much in class, but it's important to know this. They are they're like tumor suppressor proteins in a way, because they check the cell to make sure it's doing what it's supposed to and nothing's going wrong with it. And if it is, then they can actually cause the cell to kill itself. If 4 molecules of CO2 enter the Calvin cycle, how many G3Ps are produced and how many are exported? This is a 1:2 ratio. So it's 8 are made and 1 G3P is exported. Remember, during the Calvin cycle, 5 G3Ps go on to regenerate ribulose bisphosphate, and only 1 goes on to create glucose. So that's why only 1 gets exported here. If there are 40 centromeres in a cell at anaphase of mitosis, how many chromosomes will be found in each daughter cell? That would be 20, because if you remember what a diagram of anaphase looks like, when the sister chromatids are pulled apart, half of the centromeres end up on one side and the other half end up on the other side. So it's going to be half the number of centromeres in each one of the daughter cells, so essentially half the number of chromosomes as well. Which statement best describes the genetic content of 2 daughter cells in prophase 2 of meiosis? So remember, we are in prophase 2, so we already have the 2 daughter cells, and they're both haploid, but they both have 2 copies of each gene because they both had the homologous chromosomes go into each one of those daughter cells, so they still have 2 copies of each gene. So it's haploid, 2 copies of each gene. How many photons does it take to fully reduce 1 molecule of NADP to NADPH? That would be 2, because it's going to get a hydrogen, and that hydrogen is going to have an extra electron. So it's going to have 2 total electrons. Yeah, each photon represents 1 electron. What does ploidy refer to? It refers to the number of sets of matching chromosomes. So 1N haploid cells don't have any matching sets of chromosomes, whereas diploid cells do. Which of the following is a form of oxidative phosphorylation? That means that is essentially, it's another word for chemiosmosis, but oxidative phosphorylation requires oxygen, and phosphorylation refers to something getting a phosphate group added to it, and that's exactly what happens during chemiosmosis. ADP gets a phosphate added to it to become ATP. Which of the following serves as an oxidizing agent? That is NAD+, because an oxidizing agent is what oxidizes something. It's what accepts an electron, and NAD+ accepts an electron to become NADH. Enzymes bind to which of the following? Enzymes bind to substrates. Grass is green because it's green because it reflects green light. Remember, whenever you look at something, it is actually every color other than what you actually see coming into your eyes. When you look at something that's orange, it's actually reflecting orange light because that's what's entering your eyes. That's why you perceive it to be orange. So yeah, it reflects green light. Which of the following details a catabolic reaction used by animals? So catabolic means it releases energy, and yeah, this one This B answer refers to the breakdown of glucose. During what phase are chromosomes aligned up in the center of the cell? That is metaphase. Comes right before anaphase. Which of the following could be considered a cofactor? That would be magnesium. Remember, the vitamins are considered coenzymes, but magnesium and iron, they're cofactors. Which statement describes carbon fixation? It is the conversion of CO2 into an organic compound. This is what happens in plants. Plants fixate that carbon into themselves. The mitotic spindles arise from what cell structure? They come from the centrosomes. Those are the pasta-looking organelles that spew out the microtubules. The G2 checkpoint makes sure that, ooh, this one's hard for me. Don't actually, so G2, let's reason through this. G2 comes right after the synthesis of DNA. So it's not, the cell isn't cancerous, it's not this one, and this one's the M checkpoint, so it has to be D. And that makes sense because it wants to make sure that the S phase duplicated the DNA properly. During cell division, a cell plate is visible. What phase is the cell in? So in plant cells, they have a cell plate. In animal cells, they have a cleavage furrow during cytokinesis. So it'd actually be a plant cell because it has a cell plate. And it's during cytokinesis that it is visible. Which of the following has the greatest value of delta G? Delta G refers to the change in free energy. So which one of these changes the free energy the most? Well, the complete breakdown of glucose is the answer because glucose is a very energy-rich molecule, and when it's broken down completely, its products have a lot less energy. The other ones, yeah, delta G is changing, but not nearly as much as the complete breakdown of one glucose. What two products result from photosynthesis? Okay, so in photosynthesis, we're trying to create sugar or glucose to use in cellular respiration in the plant. So we know it has to have sugar as a product, and oxygen is released from plants, and they actually take in carbon dioxide. Carbon dioxide is a reactant, so it's sugars and carbohydrates and oxygen. Substrate level phosphorylation produces what percentage of ATP in cellular respiration? It actually only produces 10%. Remember, most of it comes from oxidative phosphorylation. So substrate level phosphorylation would be like glycolysis, which we know only produces like 2 ATP net. Which of the following are used in the anabolic pathway to create glucose in plants? That would be NADPH and ATP. They are used in the Calvin cycle to create glyceraldehyde 3-phosphate, which will be turned into glucose. So yeah, it needs a lot of NADPH and ATP, which it gets from the light reactions in the thylakoid. The citric acid cycle produces what percentage of ATP from glucose? Just 10%. Remember, most of it comes from the electron transport chain and chemiosmosis. Oxidative phosphorylation. Citric acid cycle does not produce much ATP. I think it only produces 1 ATP per pyruvate, or rather acetyl-CoA that goes into the citric acid cycle. So yeah, it does not produce much ATP. Which phase lasts the longest? That would be the interphase. Remember, cells, most of the time, they are not dividing, they're in interphase. Replication, that's not a name of a phase, that's nothing. So it's interphase. ATP synthase in the thylakoids. Okay, so this is important distinction. ATP synthase in plant cells takes hydrogen from inside the thylakoid, accepts it, uses its chemical gradient to phosphorylate ADP into ATP and spits it out into the thylakoid, or sorry, into the stroma. So it accepts hydrogen ions from the thylakoid lumen and releases them into the stroma. Whereas in cellular respiration and the electron transport chain, the opposite happens. It takes in hydrogen ions from the intermembrane or intermitochondrial space, and it goes down ATP synthase into the mitochondrial matrix. So yeah, it's the opposite in the thylakoids. It goes from inside out rather than outside in. Predict the end result if a chloroplast's light-dependent enzymes developed a mutation that prevented them, or sorry, light-independent enzymes developed a mutation that prevented them from activating in response to light. So this would mean the Calvin cycle was interrupted. And when the Calvin cycle was interrupted, we're going to have ATP and NADPH build up because that's what's used in the Calvin cycle to create glucose. If a organism's gametes had 17 chromosomes, how many would it have? How many would its somatic cells have? Twice as many, because the gametes are 1N, somatic cells would be 2N. Which of the following molecules is likely to have the most potential energy? That would be sucrose, because sucrose is a disaccharide, so it's going to have the most, and then glucose would have the most after that, but sucrose would have like twice as much as glucose. Light-dependent reactions in the photosynthesis occur in the thylakoids, because that's where the chlorophyll is, and yeah, it occurs in the thylakoids. The light-independent reactions would occur in the stroma. How do cells at the completion of meiosis compare with cells that are in prophase of meiosis 1? So at the completion of meiosis, you have a haploid cell, and you have 4 of them. And in prophase 1 of meiosis, you're gonna have to have all of the DNA that's going to be in those 4 cells. So those 4 cells are going to have 1/4 the amount of DNA each. And since the ploidy number goes from 2N to 1N, they're going to have half the number of chromosomes. So they're going to have half the number of chromosomes and 1/4 the amount of DNA. The citric acid cycle always returns to form what molecule? Remember, it's a cycle, so it has to come back around. And it actually comes back around to form oxaloacetate. Remember, pyruvate is what goes into the cycle. And it gets converted into citrate. But it all comes back around to oxaloacetate. What is the high energy unstable state of being that a molecule undergoes during a reaction? It's this transition state. That's where it is during the humpy. All the following are products of reduction except, well, FADH has been reduced, NADH has already been reduced, H2O is reduced because at the end of the electron transport chain, oxygen gets reduced with the electrons to form water, so the only thing that isn't a product of reduction is glucose. In the cells of many eukaryotic species, the nuclear envelope has to disappear to permit which of the following events in the cell cycle? Well, those microtubules can't exactly connect to the kinetochores until the nuclear envelope is gone, so that would be B, connection of microtubules to the kinetochores, because that has to happen. Breakdown of cohesin signals what phase? It's anaphase, because cohesin is what holding those sister chromatids together. When cohesin is broken down, they split apart, and that happens in anaphase. What group of proteins regulates the cell cycle? Well, cyclin-dependent kinases and cyclins regulate the cell cycle. However, I made this question really tricky. There's also negative regulators in there that I did not list in here, so actually it's B. I did not list all the proteins in this question, because there's negative regulators as well. Redox reactions primarily involve electrons. There are electrons being moved from one thing to another. Something that gets oxidized gets its electrons taken away. Something gets reduced, it gains those electrons. What type of cell will continue to divide without regard to space restrictions? Cancer cells, because normal cells will have an enzyme to signal when the cells are just too packed together, there's no room for division. But cancer cells, they will just keep on trying to divide. During what phase is DNA replicated? S phase. S stands for synthesis, DNA synthesis. Meiosis generally produces what kind of cells? It produces gametes. It does not produce diploid cells. It starts with a diploid cell. And it does not produce somatic cells. Somatic cells are 2N. And yeah, you would hope that it doesn't produce cancer cells either. What functional group is removed from pyruvate to form acetyl-CoA? It's a carboxyl group that's removed, and it actually goes off in the form of carbon dioxide, and during this process a NAD plus is reduced to NADH. Histone proteins have what function? Well, DNA wraps around them. Okay, that's the answer. DNA wraps around histone proteins. The electron transport chain most resembles a relay race because the electrons, get passed on from one complex to another from using the ubiquinone. So yeah, it's like a relay race. The electrons are like the baton. The final electron acceptor in the electron transport chain is oxygen, because oxygen gets reduced to form water. In a diploid cell with 5 chromosome pairs, 2n equals 10, how many sister chromatids will be found at the nucleus? Well, it has 10, so it's going to have to double that, double the number of, well, not chromosomes, but it's going to have to double the number of chromosomes in terms of how many sister chromatids it's going to have. So it's going to be double the amount of sister chromatids as chromosomes. So that's going to be 20. All the following are anaerobic except the electron transport chain. Like I just said, oxygen is the primary electron, or the final electron acceptor rather, and anaerobic means without oxygen, so yeah, it's the electron transport chain because it's aerobic, not anaerobic. Which of the following components is not used by both plants and cyanobacteria to carry out photosynthesis? So cyanobacteria, remember bacteria are prokaryotes, they don't have organelles. The chloroplast is an organelle, it's not going to have one. Which order of molecular conversions is correct for the Calvin cycle, which is the light independent cycle? So you start with ribulose bisphosphate and you add carbon dioxide to it, and the enzyme that makes this happen is rubisco. Then you get a 3-PGA, and that 3-PGA you get glyceraldehyde 3-phosphate, which becomes glucose. Which of the following is not a form of potential energy? Well, an apple has energy in its chemical bonds. A diver has potential energy that he can release by jumping off a platform. Gasoline has a lot of potential energy. Car speeding down an interstate, that's going to be kinetic energy, not potential. The part of meiosis that is most similar to mitosis, well, that's referring to the sister chromatids, which separate in mitosis. This also happens in meiosis 2, because remember in meiosis 1, the homologous chromosomes separate. In eukaryotes, glycolysis occurs in the cytosol. Remember, the Krebs cycle occurs in the mitochondrial matrix. Homologous chromosomes, well, they are not exactly the same, but they do code for the same traits. And they don't separate during meiosis 2, they separate during meiosis 1. Asexual reproduction occurs during which of the following? That would be mitosis. You don't need sex cells to form every cell in your body, and most cells in your body divide and reproduce with mitosis and form, copies of each other. Measurements of the amount of DNA per nucleus were taken on a large number of cells from a growing fungus. The measured DNA levels ranged from 3 to 6 picograms per nucleus. In which stage of the cell cycle did the nucleus contain 6 picograms of DNA? So in the G1 phase, it's going to have the least amount of DNA. During the synthesis phase, it's going to be growing its amount of DNA. In the G2 phase, it should have created all the DNA it needs for cell division. So it's going to be the G2 phase. The production of ATP from glycolysis is rate controlled via feedback inhibition. So when we have a lot of ATP, that slows down glycolysis. So yeah, we don't continuously produce energy that we don't need. So the product actually inhibits the original reaction. If no NAD+ is available during glycolysis and the environment lacks oxygen, then during the process of fermentation, we take NADH and we oxidize it back to NAD+, because NAD+ is required in the step of glycolysis that occurs before those 4 ATPs are formed. We take in NAD+ and we reduce it to NADH. But if we don't have any NAD+, we can't do that. So it stops glycolysis. But luckily we have fermentation, and we can take that NADH, oxidize it, and form lactate, or lactic acid, and yeah, glycolysis can continue. When are homologous chromosomes aligned at the equator of the spindle? So you see these Roman numerals, so we know we're talking about meiosis, and homologous chromosomes align at the center. Why did I put equation? I meant to put equator. Oops. Yeah, so that happens during metaphase 1. Remember, sister chromatids would align at metaphase 2. Unpacking of chromosomes and the formation of a new nuclear envelope is a characteristic of which stage of mitosis? That would be telophase. So right at the end of the cell division, the nuclear envelope reforms, and that phase is called telophase. How are NADPH and glyceraldehyde 3-phosphate molecules made during photosynthesis similar? They're not both produced from carbon dioxide. They're not substrates for photosynthesis. And NADPH is not an end product of photosynthesis, but they do both store energy in their chemical bonds. At what stage of meiosis are sister chromatids separated from each other? Okay, this is anaphase 2, remember. Homologous chromosomes are separated during anaphase 1, sister chromatids separated during anaphase 2. Failure at the M checkpoint could occur at what phase? That would be metaphase, because it makes sure that all the chromosomes are aligned properly. That's the purpose of the M checkpoint. So M checkpoint, metaphase, they both start with M. The process of dephosphorylation can be used to do all the following except. So dephosphorylation is the removal of a phosphate group and production of energy, which can be used to pump sodium ions, move muscles, can create glucose in terms of the Calvin cycle, and it can't produce ATP. That would be the exact opposite. Phosphorylation can produce ATP. Dephosphorylation breaks down ATP. So I am done. Let's see how I did. Yay, I got 100, of course.